here. So if I leave it like that, can you hear me? Yeah, the screen might be a good idea, shall we? Huh?
Yeah, oh, yes. I think this song already.
You mean you can't see that? I mean, what's up with you guys? I have no idea. Let's see what to do with that. Like that. Okay. What do I do with my glasses? It's not fast, is it? Huh? All right, well, good morning. I don't think this is working that well. Is this loud enough? I feel really low volume. Let's see if I put it up here. Can you hear me? Can you hear me? Yeah? Okay, great. I think I know most of you. If I don't know you, I'm Carrie Earthman. I'm professor here in nutrition, and I direct the dietetic program. So most of you I know from medical nutrition therapy, so I'm glad to see familiar faces. Dr. Chen always lets me do this set of lectures around amino acid and protein metabolism because it's something I really like, and she's just nice enough to let me come visit. And I usually try to bring some of the concepts that are relating to medical nutrition therapy and other elements of nutrition in, because protein metabolism is really, really important. So I know you all have had basic nutrition, okay, but I do go over some pretty basic stuff at the beginning, and it's mainly just to make sure that we're all on the same page and that you remember these things, because I know that you study a lot during advanced human, probably where some of this has been, and also physiology, right? So I'm going to just brace, it won't all be as simple as it is at the beginning of this, but I just want to make sure that we're all thinking about proteins, you know, and the fact is, as you are probably remembering well, the structure of a protein determines its function, right? Is that one of those mantras you learn in physiology class? And you remember that proteins have different levels of structure, right? Primary, secondary, tertiary, and quaternary. Not all proteins have quaternary structure. So I'm just going to really quickly run through this, and this is really basic, okay? So this is what you can teach your grandmother about amino acid metabolism. This is the structure of an amino acid, right? Do you remember that there's an alpha amino group and an alpha carboxyl group to every amino acid? And then each one has its own unique R group. So I'm going to show that with a circle around it whenever we are thinking about an individual amino acid. The next thing we know is that amino acids are the building blocks of proteins, right? And they come together. So here are two amino acids. The carboxyl group of the first amino acid is going to interact with the alpha amino group of another amino acid, and they're going to, in so doing, it's going to form a peptide bond and release water, right? And so here, and I'm just realized this is in red. It's not easy to see that, but this is a peptide bond that gets produced, and of course, the linkages of amino acids by peptide bonds is a complex process, right? Protein synthesis, and I think Dr. Chen is going to maybe walk you through that later, perhaps. I'm not sure if you do protein synthesis, but it's complex, requires a whole lot of energy. We're not going to focus on that. We're going to talk about all the other things around the amino acid pool and what's happening in the body once we get past this real basic stuff. So here's a summary. Here's a summary slide of what these things are, the different levels of structure for your reminding you, and then I've got a slide on each one. So this is showing the peptide bonds of a tripeptide, has two peptide bonds linking three amino acids together, right? So this would be, again, tripeptide because we've got three different R groups. That's primary structure, right? Just the linear sequence of amino acids in a peptide chain. And then secondary structure is hydrogen bonding driven, right? So the hydrogen bonds that form, they're weak linkages, but it helps to form, this is a beta pleated sheet. you know, the structure that you can imagine probably from biochem class. And there's also an alpha helix. This is also a secondary structure. So the two secondary structures, and I had that on the summary slide, are beta pleated sheet and alpha helix. And then tertiary structure results from interaction between amino acids that may not be right next to each other in a polypeptide chain, right? So can you guys think of examples of tertiary structure-driven interactions? What kind of interactions drive tertiary structure? I'm making you remember biochem, I know, because this is probably more biochem. Yeah, disulfide bonds. So those bridges that form between cysteine residues that have sulfur groups in them, disulfide bridges or disulfide bonds. Any others that you can think of? Yeah. Yeah, hydrophobic interactions. Those are super important. Because what do? They tend to aggregate, they make a protein fold up so that the hydrophobic residues are all on the inside of a protein away from the aqueous environment, which would be our body, right? Blood or wherever these proteins are. So hydrophobic interactions are really important. Are there others? What else can we think of? Yeah, please. Electrostatic, ionic interactions. Actually, we've got the weak ones, the weak van der Waals interactions are also electrostatic, right, I think. But ionic interactions, those are the ones that are probably a little more powerful than that, the positive and negative charges that tend to make the interactions between amino acids that are far away from each other. And incidentally, those negative and positive charges of amino acids are in the case of the positive charges, it's a type of amino acid. Anybody remember that? Is it basic or acidic that would be positively charged in the body? Basic, right? So basic amino acids and then acidic amino acids will interact like that with ionic interactions. Okay, so that's tertiary structure. And then the last is quaternary, and that's if there's more than one polypeptide chain. So not all proteins have multiple chains. Okay, a lot of them do. Hemoglobin is a great example. It has 2 alpha chains and 2 beta chains. And the way those interact, those 4 chains, and the way it folds up allows it to carry and bind oxygen, right? And the heme unit has iron in it, right? We've typically got 1 per chain or whatever, and so that allows hemoglobin to carry out its function. Okay, that was protein structure 101. Are there any questions about anything that I've talked about just so far? All right, it's all that's hopefully pretty clear in your minds here. Okay, so this is a roadmap of where I hope to take us in the next several days. I am not going to talk about degradation and synthesis. Again, really interesting, complex process, but it would take more than the days that we have to do that. So I'm going to start with protein digestion, and I'm just going to warn you that I have 2 kids that when they were first learning how to do PowerPoint, they have mixed up animations in this, and they're exciting animations. I'm going to tell you, they're about as exciting as the MNT ones that I have every now and then. So I think when I click, something's going to happen, but I can't remember what, so I'm just going to, yes, isn't that exciting? Oh man, okay, so we're going to start with protein digestion, okay? And if you remember, protein, you know, we have obviously exogenous, meaning what we eat, right, or what we take in. We can feed people by different routes, but anyway, that's from the environment. We'll take in protein. I've put some placeholder numbers here. Okay, this is not set for every person, obviously, but it's just an example so you can look at the proportional contribution coming from, so if you have about 100 grams coming in exogenous, 70 grams or so is coming from endogenous sources, which is the sloughed GI tract cells, digestive secretions, mucus, etc. And the main point I'll have here with these numbers, so about 170 coming into the GI tract for digestion, only about 10 of it goes out in the normal person. Okay, so we are extremely efficient at taking in protein and using protein. And the rest of that is going to enter the amino acid pool. Now, it doesn't mean that some of those amino acids aren't going to be broken down or whatever. We're going to talk about what happens to the pool over here. But you know, we're not losing a lot of nitrogen in the stool under normal circumstances. The other thing that I'll point out here while we're on this little piece is that we also get a fair amount of nitrogen from the colon from urea. I'm going to make that very strange pronunciation again. Urea, okay, from bacterial breakdown of urea. Okay, so we're not absorbing amino acids from the colon, right? That would be wildly new to you. I'm not telling you that. We can't absorb, you know, we don't absorb protein from the colon. But we do recycle a fair amount of nitrogen as ammonia getting released from urea, from that, because we have huge microbial counts in the colon. And the ones that have this enzyme called urease can break down urea. So we get a little bit of nitrogen from that as well. And that's important, those of you that are in MNT, because I'm going to bring it up when we talk about liver failure, okay? Because that's a very important source of ammonia when the liver is that we can reduce, okay? We can take strategies to reduce that. That's aside, okay? Don't worry about that, those of you that are not in MNT. But anyway. Protein losses from the body are over here, skin, hair, dermal, sweat. We get rid of some nitrogen as urea in the sweat, and then we got GI secretions, et cetera, that are going out. Those are typically a very small amount of nitrogen that gets lost that way. Okay, so let's talk about protein digestion and absorption. Very brief review, and I know this is also hopefully totally a review for you. Okay. But again, important to think about the fact that you need 3 functioning healthy organs in order to carry out normal protein digestion and absorption. The stomach, the small intestine, and the pancreas, okay? And so I'm just going to try to help us remember. So the first thing that happens, you've got 2 key elements, right? HCl and pepsin. So let me back up a moment and just say that, you know, if you remember, protein entering the stomach, right? You eat something. The gastric mucosa secrete gastrin. Hopefully you remember that. And gastrin is the thing that stimulates HCl release, right? Hydrochloric acid. It also stimulates the release of pepsinogen, which I don't have listed here, okay? So gastrin is that stimulus to release hydrochloric acid and pepsinogen, 2 different types of gastric cells. We won't worry about the names of those. So the first question to you is, what does the HCl do for protein digestion? What do you remember about HCl? And we're talking protein digestion, so we'll focus there. Yeah. Yes, that is one of the most important things it does, for sure. It activates pepsinogen to pepsin, right? So pepsinogen is what we would call a proenzyme. It's in its inactive form, and the reaction or the acidic environment stimulates autocleavage to pepsin. Okay, that's one of the things. You think of anything else that the HCl does? Yes. Shiran, right? Okay, you are, so it changes the shape of the protein. What is happening? Somebody else. Yeah, Lindsay. Okay, what does that mean to you? Did I see another hand, maybe? Or do you want to, Emily, you want to? Okay, so you're all 3 right. It is an important thing that it unfolds the protein. And it does that. Can anyone explain a little further as to what that is, how it's happening? What's the acid doing? Remember? Yeah. So that's a big part of what it's doing when we say denaturing, is it's changing the charges on select amino acids. We're going to get more into that in just a moment. And the process is called denaturation, right? What does the pepsin do? What is pepsin? biochemically speaking? It's an enzyme, absolutely. And what does that enzyme do? Yeah, breaks the peptide bond, right? So pepsin is kind of the first attack on that protein. Well, the acid is kind of the first because you have to unfold it so that the pepsin can get in, right? And it's an endopeptidase, so it cleaves amino acids from somewhere within the chain, the unraveled primary sequence chain. I'm going to back up to HCl one more time just to make sure we're clear about this. Does the HCl do anything to the peptide bonds? I'm seeing shaking heads, right? Can everyone recall that? That's an important thing to remember. Acid does nothing to a peptide bond. It's a very strong covalent bond, right? You need an enzyme to break it. So HCl does nothing for peptide bonds. It does everything to unload or to unravel the structure of the protein so that pepsin has free access to start chopping up that protein. Incidentally, HCl does another thing for us in terms of food safety. What does it do? Yeah, it kills bacteria. Why? What is the pH of the stomach, roughly? Yeah, 1 or 2, 1.5 to 2.5 typically. You'll see ranges. It differs, obviously, a little bit person to person. So HCl is a very powerful protective device for us. Helps us go to the state fair. Okay, so mainly what's leaving the stomach are really large polypeptides and oligopeptides. I mean, you'll have some free amino acids, but mostly you're talking about big peptides. So it's not a ton of digestion, but it's important, really important first steps that happen in the stomach. The next bit, and I'm just going to make sure I'm following my other notes here. Next step that happens is the small intestine, and protein digestion finishes there, where proteins are digested by the combined action of intestinal proteases and the pancreatic proteases, right? Those that are released, I'll click here. Combination of exo and endopeptidases, right? So you can chop amino acids off typically from the amino end, but they also go in from the carboxyl end, different ones. Or the ones that are endo means they're chopping the oligopeptides into tripeptides and in the middle of the chain. And they are often specific for certain amino acid residues, but they don't have to be. So there's a mix of proteins and enzymes that are working together to get the protein digested. Now we're going to kind of walk into this. This is another really basic. Wow, that's huge too from here. Yay, look at the pancreas. So you remember that pancreatobiliary secretions, pancreas biliary combination secretions, they're entering in at the duodenum, right? Again, important to remember this physiology for MNT because when we talk about removing certain parts, you have to think about that. But it's really critical that the gallbladder, the liver, the pancreas, all are secreting the various aspects to help facilitate the process. And incidentally, I know you will remember this. What is the pancreas? What is one of the things besides pancreatic enzymes? What else does the pancreas secrete to really facilitate digestion? Yeah. Well, that's true, actually. You know what? I wasn't thinking about the endocrine functions of the pancreas, but insulin is important, but not for the moment. Put that aside, right? Really critical, but not for this particular thing. Yes? Bicarbonate, yes. Bicarbonate, right? And what is bicarbonate doing in the duodenum? It's raising the pH, right? And what does that do to facilitate digestion? Say it louder, would you please? Yeah. Well, the bicarb does not activate the, but that has to happen. So hold that thought. Yes. It does change the pH, right? Okay. So acidifying or neutralizing the acid chyme coming from the stomach is important, right? What role does that play in optimizing digestion to increase the pH of the duodenum?
Break down the enzymes or anything?
Not break down the enzymes. Is that what you said? Not quite. Anyone else? Yes. So that's where biochem comes in, right? Activating the enzymes, making sure they're at optimal pH for optimal activity. And so that allows for optimal protein digestion, right? Okay, and here's I'm just walking through it in a little bit more detail real quick because I think we hit most of these. So you've got the acid chyme coming in from the stomach into the duodenum, and it's containing these various oligopolypeptides, some amino acids, goes to the intestinal endocrine cell, and the first thing happens is CCK, cholecystokinin, and secretin are released from the intestinal endocrine cell, right? And so that's the first really important mode of action of how all this happens, right? That release of CCK and secretin acts on the pancreatic acinar cells, right, to release trypsinogen and bicarbonate. Okay, and there are other enzymes that the pancreas is going to release that are animated. You already got them on your slide, right? So that's the first important thing. And is they're acting, they're carried through the blood, go to the pancreas, bicarb gets released, trypsinogen gets released, right? CCK and secretin also act locally in the intestinal epithelial cell to release this really central enzyme called enteropeptidase. Have you heard of that one? Yes? Good. I'm glad. Checking my colleagues here. So enteropeptidase is the enzyme that acts on the proenzyme trypsinogen, or zymogen. You know that term? Right? Zymogen is the same thing as a proenzyme to activate trypsin. And then once some trypsin is made, trypsin can then auto-catalyze, auto-activate other trypsinogen. Oops, sorry, gosh, that was overly fast. Move it back. So trypsin will act on more trypsinogen to make more trypsin, and trypsin is the key to activating these other proenzymes. But you have to back up. Without enteropeptidase, you're not getting any or very little protein digestion at this level because you wouldn't be able to activate the pancreatic enzymes. Okay. And we talked about what the bicarb does. So important to neutralize the acid chyme and raise the pH, and that is all about making those zymogens, those when they get activated, make them be optimally active, right? So they can really break down the protein. Okay, now we're going to move into, yeah, please.
Without the enteropeptidase, you will not be able to activate the enzymes in
The pancreatic protein digestive enzyme. So without enteropeptidase, you can't activate the pancreatic enzymes. Yep. All right, so the next diagram is pretty basic as well. We're going to start, if I can get it to go. So up here, we're in the stomach. We got what enters the duodenum are these larger polypeptides and some smaller peptides, right? We got the pancreatic proteases, and then we have these intestinal proteases, the amino di and tripeptidases. And there are lots of them. The mucosal, you know, these are going to be acting in the lumen of the small intestine. And let me just click because I'm not totally sure where I go from here. Yeah. So on the brush border, you know, the microvilli of the small intestine, we've got lots of aminopeptidases or basically small aminopeptidases that will break down little peptides into smaller peptides. I've lost my notes. Let me just look and see what exactly I want to say here. Yeah. So these aminopeptidases tend to be exopeptidases, and they will allow for basically what's entering into the enterocytes are amino acids and small peptides, and then there are intracellular peptidases that will work on at least many of these small peptides to release free amino acids. Now we also, I think I need to go to the next slide here to get closer to the brush border, okay? We have separate, different sets of transporters and separate absorption mechanisms for small peptides and free amino acids, okay? And so that when we talk about transporters, we're talking about active transport. So small peptides can come into the enterocyte as well as free amino acids. So first point, let me just look at my list here. First point is there are separate absorption mechanisms for small peptides and amino acids. Second point is actually the primary way that amino acids get absorbed is as small peptides. Okay, as they come across, there are more of the, or more, proportionally more are coming in as these small peptides into the enterocyte through those small peptide types of transporters, active transport. Then when they get in, many of them are going to be hydrolyzed to free amino acids. Free amino acids, particularly after a meal, are going to enter primarily through diffusion, both simple and facilitated diffusion, to cross the basolateral membrane of the enterocytes and into the blood. Okay. So I said that they enter via diffusion, and then I wanted to say that we now know that there are some small peptide transporters at the level of the basolateral membrane, so right next to the capillary, you know, the interface there, that allows select small peptides to cross into the blood. And a lot of that, well, many of my colleagues, or at least a few of my colleagues around here, are looking at that. I did a study on soy-derived small peptides that have physiologic functions. So there are peptides that come from whey, for example, that are thought to lower blood pressure, for example. So this is kind of a little bit of a new area in terms of how exactly that happens. But it's important. So it's important that and they're typically small peptides, like 2 or 3 peptide, 2 or 3 amino acid, and not always. They can be, I think maybe less than 10 would be a safe bet. These small peptides that can come from various proteins and have biological action. So they're called bioactive peptides, if you see the literature, or physically functional, physiologically functional. And so remember again that I said that the main point here, because I've said a lot of things, is that peptide active transport into the enterocytes, that is the primary absorption mechanism for amino acids. It is also faster, typically faster absorption from small peptides than the free amino acids. And there are some reasons for that, okay? And I've got some, I think it comes soon, where I'll talk about the characteristics of transport. This is another important point. We're going to get to the liver soon, okay? So just remember that as those amino acids, after a meal, they're entering the bloodstream through facilitated and simple diffusion, they're going to be taken up by the portal vein into the liver, and that's kind of the first pass metabolism that we're going to talk about. So after a meal, a lot of those amino acids are going to be taken up into the liver, and we're going to talk about what happens there. Okay, so characteristics of amino acid transporters, okay, not peptide transporters, but amino acid transporters, is there is redundancy, so that's a good thing. Means we have more than one transporter to carry a particular amino acid. And then the other important concept is competition. So a transporter for 2 amino acids, those if you have more, so say example, and I'm not going to, don't quote me that this is the correct thing, okay? I'm going to say a particular transporter transports both glutamine and asparagine. I suspect that's true, but I'm not totally sure of that because they're cousins. If you have a whole lot of glutamine in the system, you're going to beat out the asparagine, right? The glutamine is going to kind of compete. So that's the concept there for the same transporter. Each tissue has a unique complement. It doesn't mean that they're all totally unique to each tissue, but particular tissues that need a particular type of an amino acid are going to be more likely to have more of those transporters. So muscle has more of the branched chain amino acid specific transporters because muscle tissue does a lot with branched chain, right? And then their last concept is transporter activity is affected by the hormonal milieu and the physiologic state. And the best example I have for you here is that glucagon will increase the A transporter, the carrier of alanine, into liver cells. And I'm just going to ask you a basic question here because I know you know the answer to this. Why would you think that would be? Why would glucagon increase the A transporter that is select for alanine transport in the liver, getting alanine in? What do you think? What's the liver using alanine for? Let's start with this. What physiologic state is glucagon high compared to insulin? Fasting? More global. Sorry? Not so complex. Real basic. Is alanine, what, please? Sarah, is that Sarah way up there? Yeah, because under fasting conditions, you're wanting to make glucose from non-carbohydrate sources, right? And so under those conditions, the liver will bring in more alanine, so it's got that precursor for gluconeogenesis. That make sense? I'm sorry, I got two sets of notes. I was trying to work them over, and I realized it's confusing. So I said an amino acid may be transported by a number of different transporters, right? I said that, and I said amino acids for the same carrier compete with each other, and there's a lot of overlap. Yeah, I think I covered that. Okay, so next final on characteristics around amino acid absorption is that the characteristics or the absorption characteristics of different categories or types of amino acids, absorption characteristics will vary, okay? Essential amino acids tend to be absorbed faster than non-essential. Do you know that language, or do you think about dispensable and indispensable? Okay, yeah, that's how I learned it, so good. I try to, I know some of the, like I think Stepanek talks about dispensable and indispensable. I'll use both so you, when we get to that. But so essential are absorbed faster than non-essential. And large amino acids tend to be absorbed faster than small ones. And don't ask me to explain why. I pulled some of the old papers from, a lot of this work was done a long time ago. But that is, I think that is still considered to be true. It's not super, critical, but. And then we've just learned that amino acids for the same carrier compete, right? So I think I took it off of your slide. Whoops, that's a different one. Let me ask you this question first. So if you had a friend who is interested in bodybuilding and you find out is taking a supplement that is rich in glutamine, leucine, maybe 2 or 3 amino acids, maybe arginine. I mean, there are these supplements out there that might have 2 or 3 only amino acids. And he or she is, taking this twice a day, plugging the nose, gagging it down. Have you ever had them, these solutions? They tend to be pretty darn bitter. Free amino acids are quite bitter. So clinically speaking, that raises challenges for feeding people elemental feedings that are just free amino acids like Vivanex. You put Rosie's lime juice, you put sherbet, you're doing everything to try to mask that bitterness. So anyway, and they're spending a lot of their hard-earned money on their college budget taking these. So turn to your neighbor, and I want you to talk about, and with these concepts in mind, what would you tell that person in terms of, is this a good thing to do? to stimulate, optimizing their muscle anabolism, or, why or why not, and what's the argument for or against? If you, come down for, then explain why you think it's for, and vice versa. So turn to your, if you have hopefully you have all, you're sitting close enough, hopefully. It can be groups of 3 if it's easier, okay? Just talk for a moment. Here. Okay, I heard some people talking about the question, others were talking about MLK Day. No, I'm giving you a hard time. So hopefully, what did you come up with? So how many said that you are in support of having this person take the supplement that has the 2 or 3 amino acids? Okay, so was that a rhetorical question, perhaps? Okay, so we say, okay, we don't think it's a good idea. So why not? What arguments did you all talk about together? Anything we can talk about? Yes. Okay. Yep. Okay. Yep. So I'm going to repeat what Anna just said. So by giving just a few amino acids, you're going to potentially cause competitive inhibition at the transport level, so that maybe you might even create an imbalance, right? It's certainly possible. Were there any other? Yes. Yes. Yeah, most likely. Yeah. So, I was just talking down here. The kind of studies that are available are not, they're usually the really clean, where you have nothing coming in. You do whole protein versus, protein with or amino acid supplementation or whatever. They're not perfectly great. So a lot of this is what we understand about the mechanisms. And when you look at whole anabolism, it's less when you have this potential for competition, when you don't have a lot of other sources of protein coming in. What was the other one that we talked about? I just totally lost my train of thought. So it was, right. So not only can you have competition, was there something else that you know about optimal protein digestion that free amino acids wouldn't meet? Yeah.
Well, just like if you have a free amino acids, they don't get absorbed as fast. It's like maybe the flow.
Right. So you're not optimizing efficiency of protein assimilation when you're just giving free amino acids, right? Because peptide transport, we know, is actually more efficient and better. It doesn't mean that you can't eat free amino acids, but mostly what we're eating is whole protein, right? So I hope physiologically this makes sense, right? I mean, if you think about it, we didn't evolve, to be consuming crystalline amino acids. So it's important that we're able to digest whole protein and peptides. And the other piece of it is, these things are expensive and they taste bad too. So, you can really save your friend a lot of heartache if you just, apply these various things. Concepts. I'm not speaking very well this morning. So it seems to not want to advance. Why not? Let's see. Oh shucks, an escape doesn't work.
Yes.
So I'm gonna move on. You guys have a slide, I'm pretty sure, a little case scenario. You have that? Oh, thank you. Are you a miracle worker? Yes, you are. Yay, thank you. Okay, so we have a little case scenario here. Very simple. Obviously, I'm not giving you any information really, except that the patient has pancreatic insufficiency, right? So mild suppression, the pancreas is not working super well. They could have chronic pancreatitis and have really significant pancreatic insufficiency, or they could have cystic fibrosis or some other problem like that. So clinically, we apply these concepts every day when we think about formula selection for people that have particular problems, right? And so I could have said somebody who had a short gut, right, who has very limited absorptive capacity. That would be another possibility. We could also talk about, is there anything we need to do differently if somebody has their stomach removed? And the answer to that is probably not, because we get so much good protein digestion happening in the intestine that, we're pretty good if we have a small intestine. But the kinds of formulas that are available, which one do you think would be best for somebody who has pancreatic insufficiency? And we're just thinking about the protein, right? In MNT, we'll talk about the whole formula, but just the elements of the protein. So standard polymeric, that's like eating a hamburger or something. It's not, of course, in a tube, but similar, whole protein kind of a concept. The second one is partially hydrolyzed. It's peptide-based, mix of small peptides and free amino acids. And then the third one is completely hydrolyzed elemental types of formula. So show of hands, who thinks it should be A, B, or C? Okay, may I ask you why you think C? What do you, why would you recommend that? Where's your thought? Okay. Okay. And so I'm not to put you on the spot, just to ask so I can make sure that we're really clear about when we talk about, so Katarina's concerned about the fact that we don't have adequate pancreatic enzymes, which is a totally viable concern, right? So let me maybe back up a moment. First of all, to say that when we say pancreatic insufficiency, it doesn't mean no pancreatic activity, okay? I should have maybe made that clear. So we can assume there's some, okay, but it's diminished, right? So an important point you raise when you think about that, and I'll just say, was there any other thing that anyone else thought that maybe an elemental would be better that I'm not addressing? So the intestinal enzymes are still active, okay, and pancreatic insufficiency is something that again, is not a complete shutdown. So you'll have, and when, and this is where it's always hard when you pull something out like this, because pancreatic insufficiency, bicarbonate production is pretty darn voluminous, right? So you have to have pretty much no pancreas to have no bicarb. So you get some produced. Now we can do things to facilitate the digestion as well, okay? So I was thinking down the lines of amino acid absorption mechanisms, and not so much about, so thank you for making me think about the physiological, what I've set you up to think about. So if you have modest depression of pancreatic action, still have some bicarb, you have some pancreatic enzyme activity. Most of you chose this one, Was that because of what we talked about in terms of amino acid absorption? And I would say that's very where I want you to be, because you want to think about the fact that you're optimizing those mechanisms, the transporters for small peptides and amino acid transport, and basically can facilitate. The other thing we do, and this will support Katarina's thinking because it is important, okay, is that if somebody has significant chronic pancreatitis, for example, where we start to see malabsorption and fat in the stool, et cetera, we give pancreatic enzymes, and we also give antacids, so that we're basically raising that pH and optimizing what activity there is from the pancreas, okay? So, but even with all that, we now know that peptide-based formulas are really where we want to be. I give you all the formulary from Fairview in MNT, and it's got Vivanex on there. Vivanex is the elemental formula that I grew up on when I mean, not me personally, but as a dietitian, I was using that all the time in my inflammatory bowel patients. And now what we know is that actually it tastes really terrible, so if you're trying to give it to somebody orally, it's horrible. And peptide-based formulas are better absorbed, and they taste better. So there you go. And that's what we do now for people that are really having mild or even moderate pancreatic insufficiency. Any questions about that? Appreciate you all being so awake this morning. Okay, and there's our summary. I don't think I need to revisit that. It is 30, and we are at the end of a section, so why don't we take a break? I know I give really short breaks in MNT, but Dr. Chen is kinder to you guys, so I'm mean, aren't I? I mean, and it's at night. If those of you that are not in the class, you can all have a collective aww, you know, for people that are in MNT. Although I'll remind you, it's not on Friday, right? Okay, so you have 8 minutes. How about 8 to 10, something like that? I can't even see the clock. Is it 28 after? Come back by 20 till maybe.
Yeah, I mean,
Yeah, because it might go.